Good morning, everyone. This is Adam Bianchi at the Accreditation Council for Medical Affairs. I am pleased to have everyone here this morning for what I think is going to be a great discussion of an up-to-the-minute scientific view of treatment, prevention, and management of COVID-19. And we're going to be taking a look at how medical affairs has played a role, the impact on medical affairs, and this we have the series of webinars where we're exploring these topics. Very honored to have Dr. Naji Al-Sayed with us this morning. He's the former Global Medical Director at Merck, and is currently a partner with Cogent Healthcare. Before we get started with Dr. Al-Sayed's presentation, Dr. Christine Magala is going to update us on the COVID-19 task force that ACMA has set up and some of our current activities. So please welcome Dr. Christine Magala.
Thank you, Adam. Thank you, everyone who is joining us today. So I just wanted to provide a few updates on a few of the initiatives that the ACMA is taking. So the ACMA has convened a COVID-19 task force, which is made up of leaders from the industry, academia, and medicine. And we've put together a COVID-19 resource page, which we update on a daily basis to bring you the most up-to-date information about the pandemic. We know that financial hardship can be a challenge now for many. We definitely don't want that to be a barrier to becoming board certified in medical affairs because we know how important your professional development is to you. So we are also providing financial assistance and various payment plan options to help individuals who want to pursue the board certification. We also, on the COVID-19 resource page, have a free COVID-19 course that is available for everyone as an additional resource. I would definitely encourage you to check it out. And we are also very excited to announce that for leaders at pharma, biotech, or medical device companies who want to board certify their MSL medical affairs teams during the pandemic, the ACMA will take a portion of the proceeds and donate it to hospitals and healthcare workers that are really struggling right now to help provide them with the much needed resources to help them while fighting the virus on the front line. With people being out of work, many don't know where their next meal is going to be coming from. So we are currently raising money to donate to food banks and pantries that are quickly becoming depleted in the most devastated areas. If anyone has any questions about any of our initiatives or our programs, please feel free to reach out to us. We know these are extremely tough times, and we want to remind everyone that we are here and ready to support you in any way that we can. And we hope that you and your families are staying safe and healthy. And with that, I'll turn it back over to you, Adam.
Thank you, Christine. So I want to welcome our main speaker today, Dr. Naji Al-Sayed. As I said, he is a partner at Cogent Healthcare, and he was formerly a global medical director at Merck. His experience in pharmaceuticals spans over 25 years with senior positions across clinical development and medical affairs. He's worked at Pfizer, Novartis, Sanofi Pasteur, Merck Sharp Dome, Cubist, and Merck. He has over a decade experience in infectious diseases, treatment, and prevention. Dr. Al-Sayed he's practiced in internal medicine and nephrology, and he was a researcher at INSERM in Paris and at the NIH in Bethesda, Maryland. And with that, welcome Dr. Al-Sayed.
Thank you, Adam, and hello, everyone. Thank you for joining the topic of the webinar today, and thanks to Acma for the kind invite. I have the pleasure and the challenging task today to review with you the latest COVID-19 pandemic development. Obviously, Can we progress to the next slide, please? Obviously, before diving into the actual presentation, I wanted to share with you the fact that, as you know, there is really an overwhelming number of publications and activities around that. And there is today over 5,000 publications in such a short 3 to 4 months time. And that actually imposes a certain number of activities. Can we have the next part, please? So the implication of this is mainly the fact that we definitively need to prioritize the information as we're getting a lot of information in a short time. We need to categorize this information, but we also need to be very careful about the quality and validity, since the fact of having such a level of activities and publication might actually impact the quality and the rigor we're having around these publications. Sometimes it can lead even to confusion, and obviously the sourcing aspects are important as we go. Next, please. So the presentation today will be articulated around mainly the COVID pandemic fundamentals. What are the critical known and unknown? Review the approaches for the management of COVID-19 pandemic, and of course, the aspect of summary and takeaway messages. Next, please. So before diving into the actual data, clearly there are three main aspects that we need to keep in mind. The virus, which is, the SARS-CoV-2, and that stands for the severe acute respiratory syndrome coronavirus 2, and I will come back to why it's called like that in the next slide. The disease, which is the coronavirus 2, and the pandemic, and that's the fact that this actually virus and disease has spread out worldwide, and that's why it's called COVID-19 pandemic. Sorry for the actually typo. This is not COVID-192. This is COVID-19, and the two refer to the reference. Next, please. So that actually, the fact that you have these pandemic aspect impose a multidisciplinary management and measures that goes not only to the individual patient management as usual when you have a disease and a virus infection, but also to the pandemic aspect that is managed at a much larger level with much more key players having to be integrated in that. Next, please. Now, the virus is, as you know, an RNA virus that was luckily very quickly sequenced, which allowed us to have the real-time PCR testing, which, as you know, was absolutely fundamental to be able to test the patients and confirm that they are infected with the the SARS-CoV-2. The fact that there is an 80% actually genomic identity comparability between the SARS-CoV-2 and the SARS-CoV, which, as you probably already know, has been implicated in a pandemic that spread out in 2002 and 2003, explain why this one was called SARS-CoV-2. Now, the main thing that we need to keep in mind that is, since we have a relatively long incubation period, 5 to 7 days, it seems that during that period, especially at the end of that period, patients are potentially contaminating to others, which, as you can imagine, poses a problem in terms of disease spread and viral spread. The preliminary estimate for the R0 is 2 to 3, which means that once again, the spread out of the disease is relatively fast. Next, please. So if we go to the disease part, clearly the epidemiology is moving, unfortunately, fast and increasing. As we talked yesterday, we were at 1,900,000 something, and now we are at over 2 million confirmed cases worldwide, with unfortunately a large number of deaths, but luckily also over 500,000 now that have been recovering. The disease has 3 sequential stages. As we said, the asymptomatic one and the early, relatively mild one are the one that comes first, of course. But then, unfortunately, comes the 3rd phase, which is not the case for all the patients, luckily, but it happens, and this is where these pulmonary complication comes in, and we will come back to this in a second. The most frequent initial symptoms are the fever, and that's important because, as you know, this is so far one of the methods that we are using to screen the patients and potentially detect those who are maybe asymptomatic but having some fever or mild symptoms. The cough is an important one, and the fatigue are the 2 other very predominant symptoms of that disease. Now, moving to the severe complication, often these severe pulmonary complications comes around the 10th day. So it is not happening immediately as the patient's being contaminated and become symptomatic, but unfortunately, around 10 days after, some of the patients are developing pulmonary complication, which could go up to the acute respiratory distress syndrome. Now, these ARDS are actually more predominant in men versus women. It is negatively correlated to age and comorbidities. And it is important also to keep in mind that it looks like the phases of the disease are the initial adaptive immune response, which is the early stage of the disease, but then kick in as the patients, and luckily not all the patients, become more complicated and goes to the pulmonary complication. And then you have the innate inflammatory response, which obviously make the situation much more complicated to manage and often lead the patients to the ICU. Now, the other very typical aspect of the disease is that it's a really prolonged stay in the ICU. It's 2 to 3 weeks. For reference, usually a patient stays within the ICU for a week. So you can imagine that it's a really long stay in the ICU, and the survival is unfortunately not extremely high. That goes from 20 to 50%, depending on the countries and the publications. Now, the preliminary case fatality estimate is 2 to 3%, and luckily, this is still a disease where 81% of the patients that are infected actually require no hospitalization and can be recovering after that. Next slide, please. Now, I will go quickly to the critical unknown. As you probably know, this is beyond, as compared to SARS-CoV and MERS-CoV, it seems to have a much higher spread out capabilities. The shedding is not totally unknown. There are many publications, some of which I would say contradictory. The protective aspect or neutralizing capability of antibodies in terms of efficacy and in terms of duration is not fully clear yet. And even the peak is in some gray area of understanding when and how high this peak need to become. The virus mutation propensity is at question. There are some recent data that suggests that actually it doesn't mutate very quickly, which is potentially a good news if confirmed, especially when we think about vaccine development. The seasonal nature is not very clear whether this is going to be actually a seasonal virus like actually the flu, or it's going to be a different one. The actual proportion of immune population is not known today. And obviously, until we have an operating large-scale serology testing, this cannot be done. The actual case count, including the mild and asymptomatic patients, which are needed to have the true mortality prevalence, are not available today. Still a gray area is the disease and infection severity in relation to comorbidities. For instance, when we talk about obesity, obviously obesity, it can be an aggravating factor when you go to mechanic ventilation. Nevertheless, there are some data that actually are questioning whether obesity by itself, it's also a risk factor for patients to become infected and severe with the COVID infection. So the final one is obviously the most important also one, is no current vaccines or proven efficacy treatment today. And although the large-scale serum antibodies dosing being developed and seems to be soon approved, this was not available until recent day. Next slide, please. Now, going to the various approaches to the management of the pandemic. Next, please. We have articulated this around three areas: the infection control, the infection prevention, which in some cases actually can be also considered as part of the infection control, and obviously the infection treatment and specific patient management. Next, please. Now, we're all familiar, obviously, with the infection control measures in terms of confinement. I would just mention that clearly there are two different strategies that are followed according to countries. The general one, like the one is done in the US in certain of the states, in France, in Italy, in Spain, but the one that is more selective, that is, for instance, done in Sweden, and that was initially meant to be also the case in UK before they switched to the general confinement. The mask wearing is also one that follows these two aspects of selective or targeted or generalized. Now, there is a lot of debate around this, whether this was a purely medical decision or actually more by lack in some cases of availability of these masks. There are pros and cons for that, and probably you can come back to this if you wish within the discussion part. And of course, lab testing, as we said, is very important aspect when we go to infection control. Next, please. Now, I'm not going to go through all the vaccines, but what is important to keep in mind is that this is, although a moving target, and luckily more vaccine candidates are being tested and developed. There are 3 of these that are now within the clinical evaluation, and 67 within the preclinical evaluation. And the WHO have launched actually Solidarity, which is an international program to test a number of vaccine candidates at the international level. Next, please. Now, there are unknowns that are actually important and will be some of the complicating factor as we develop these vaccines. In a gray area, you have the virus mutation propensity, as we said. Whether this is going to be also potentially seasonal or not. The neutralizing aspect, whether there is an efficacy and to which extent of these developed actually antibodies, and what is the duration of that protection when it happens. actual proportion of immune population, and last but not least, the appropriate animal model to start with when you develop a vaccine, which is a classical step. And that's actually why some of the companies have moved immediately, skipped that stage and moved immediately to the developing a human. Next one, please. Now, going specifically to the infection treatment and management. Next, please. Now, clearly, if we look at the MERS, there has been obviously significant development covering area of diagnosis, prevention, i.e. vaccination, and therapeutic area. But giving the more regional spread, the amount of research was important, but yet nothing to do with the SARS one. Next, please. And as you can see here, there is a much larger number in every one of these areas, from diagnosis to prevention and to treatment, that are developed and patents that are being actually acquired. And that's not to say that it's only a patenting aspect. When you move to the development, the actual development part, next please, you can see that you have also a number of these that were developed across the whole spectrum, from diagnosis to treatment and prevention. Next one, please. So I think it is important to look at how these development strategies are done through the spectrum of three pillars. The first one is the repurposing of already approved drugs. That's obviously the quickest development, because we know these drugs, we know the safety, we know the efficacious dosage, although that might need adjustment, obviously, when you change the type of a disease that you are targeting with such a drug. And then the second one is, of course, the in development compound screening, with a priority to those that were actually initially developed to target SARS and MERS candidates, and the new candidate and therapeutic approaches as also one of the much, I would say, more midterm hope. Next slide, please. Now, once again, I'm not going to go through all of these compounds, but clearly there are 9 major compounds that have been tested for the treatment, and all of these were repurposed as they were already approved a drug, as you can see from various other type of diseases. Obviously, what is important is to see that most of these are antiviral drugs, but also some has activities as immuno-inflammatory modulator, giving what we mentioned about this phase of immunoinflammation when the pulmonary complication stage kick in. Next slide, please. Now, I'm going to go quickly through the various programs that are actually involved today, and take the main one. There is the pan-European one called Discovery. The objective is obviously to look at the efficacy and safety of these 4 experimental therapeutic. The first option is remdesivir. The second one is a combination of leponivir and ritonavir, and without using interferon. And the third one is the addition of interferon. And there is a 4th arm, which is the hydroxychloroquine. Now, the large number of patients, of course, justified by the fact that we have these 4 arms, but also to speed up the inclusion, 8 European countries were involved. Now, this is a multi-center study, but it has an adaptive design, which it means that if we notice that one of these options either is not delivering any positive results or showing superiority to others, we can change and then switch the patient. Nevertheless, these are actually not double-blind studies, and they are conducted against the standards of care in these countries. The efficacy is done 15 days after. Next slide, please. And the Solidarity program, which is run by the WHO, is similar to actually the one we have seen from Discovery in terms of design. It's a larger one, it's more international, without the participation of US and European countries, with several thousand of patients in over 90 countries. Of course, the same efficacy evaluation after and safety is done after 15 days. Next, please. Now, moving to the US, I choose actually to kind of show the two main aspects of the development. The first one, which is the remdesivir also study in COVID-19 patients. It is led by the NIH and managed by the University of Nebraska. It obviously evaluates the safety and efficacy of remdesivir in hospitalized adult patients, so similar to the others. But the difference is that it's a double-blind randomized clinical trials versus placebo. The initial number of patients is 100 patients that are unconsidered, which can be adjusted obviously as interim analysis can take place. The other studies that are also conducted and programmed in the US is the hydroxychloroquine and the chloroquine one. The plasma therapy, which basically consists of drawing blood from patients who relatively recently recovered. Generally, we believe the peak is achieved of antibodies achieved at 7 to 10 days and after the end of symptoms. And the antibodies actually are isolated in the serum, and these are used to help the most severe patient. Now, this has been actually used. It's not a fully new technique. It has been used in other infective viral diseases before some treatment were available. And finally, there is the development of a vaccine, which, as I mentioned, entered already into phase 1 with 45 healthy adults and already started enrollment. Next slide, please. The other programs are the hydroxychloroquine and the chloroquine one, with 2 studies being initiated in France, one by Professor Raoult, with over 1,000 patients, but unfortunately no direct comparative group, which poses a certain questions to the validity of the outcome, especially when we remember that 80% of these patients luckily still recover from that disease, and that this target of the test in the Professor Raoult studies is the early stage patients actually, and not the complicated one. There is another one also run by Professor Dubé in France, which enrolled 1,300 patients and just started 10 days ago. There are several actually series studies, but coming from China, which have been published, but they are really, and to some extent, contradictory, and from a methodological point, a little bit suboptimal. Sweden has started to use this, but choose to stop it because of the cardiac adverse events, and Italy and France, as we said, are still evaluating that option. And now the other approaches in terms of clinical trials is one with a plasma therapy, similar to the one in the US that is run by Professor Lacombe in France. And there is one that was targeting more the symptomatic actually handling of the patients that goes into the phase of respiratory complication and mechanical assistance. In that case, actually, there were some promises with an extracellular hemoglobin oxygen transporter that was based on a kind of a worm that was found. Unfortunately, the study is currently on hold because of some safety concern in the animal model, which was the pig in that specific case. Next slide, please. Now, once again, in terms of how we develop the drug, I think it is important to see that there are 3 areas that we need to target as we develop these drugs. The first one is obviously the antiviral targeting treatment candidates. These are likely to be useful in the initial phase of the disease to stop the progress of the disease and hopefully preventing the disease from developing the most severe and life-threatening one, which are the pulmonary complication. The second one, which is the immunoinflammatory modulator, and these most likely will find their usefulness and therapeutic value within the advanced stages when patients are having pulmonary complication, and in some extent even moving to ARDS. In that specific case, these drugs will most likely be theoretically at least useful, and the studies are ongoing to prove that part. And the final one is the more symptomatic treatment of these respiratory complications, especially when patients are suffering from ARDS. Next slide, please. Now, if we look quickly to what is being developed, you can see that indeed these developments in terms of treatment are following the treatment of symptoms and the antiviral group, which both of them are, as we said, targeting this first category that we just mentioned. The second one is this anti-inflammation and immunomodulator one, which should be more helpful actually in the complicated phase when patients move to these pulmonary complications. And as you can see, when you look at the number of candidates that are being tested in this area, you almost have 50-50 of a split. And of course, we have over 30 drugs that are candidate but are still in development phases. Next slide, please. Now, moving to the summary and takeaway. Next slide, please. I tried to summarize actually the key things in 10 points. The first one is that obviously this is a huge impact, not only from a medical standpoint, but also from an economical and populational standpoint. There are, as we could see, a number of critical unknown that are remaining. The SARS-CoV-2 is still a fast-spreading virus, although not the fastest one we know about. It is mild in over 80%, which is a good thing, of course, but unfortunately has a potential for ARDS and deaths in a certain number of cases. These ARDS are clearly correlated to the gender, with higher percentage in men, higher actually implication with age, over 60, and comorbidities in one of the 2 patients. Now, the overwhelming pressure on ICU beds, resources utilization, and nursing home explain why, although this disease is actually mild in over 80% of the patient, why it is causing such a threat and complication to our healthcare system. So if you add this to the fact that, if you remember, we mentioned that the stay at the ICU is around 2 to 3 weeks. So really prolonged period. So the large number and the prolonged stay at the ICU is really putting a lot of pressure on our ICU resources. Today, the infection control is unfortunately the only proven efficacy measure, although, as we said, there is hope with a number of drug being developed and midterm vaccine. Significant R&D efforts are mainly hopefully coming on a short-term basis from this repurposing of identified already approved drug. And there are two short-term therapeutic priorities, in my view. The first one is preventing the disease progression to go to ARDS, which is obviously a purely clinical objective, but an important one, as these ARDS are often associated with this increased mortality. And the second one is the boosting of the ICU resources and optimization of ARDS management capability worldwide, so we can really manage these patients when the wave comes in. And finally, vaccine is definitely an important option, but it needs to be considered on the midterm since it's going to take a year and a half to 2 years. Thank you very much, and I'm happy to take any questions.
Thank you so much, Dr. El-Sayed. We do have a few questions that came in. The first one is, RNA vaccines seem to have low stability and low success rates. Is that true?
Well, actually, it's not fully true. There are complications, I would say, additional complication and point of prudence as you develop these. But we know that we can develop these RNA actually vaccines, and there are several examples of that.
Thank you. Another question is, what is the risk of reinfection for patients who have successfully been discharged from the hospital after treatment with hydroxychloroquine and azithromycin therapy? And what is the risk that these patients would still be able to spread COVID to the public?
Yeah, it's actually two important questions. The fact that these patients are discharged from the hospital once they recover is not actually dependent on the type of treatment they received or not. And therefore, when they recover and they are discharged from the hospital, there are reports saying that they still actually potentially contaminating for a couple of days, but these are sporadic reports. The risk for these patients to get infected again, once again, it's not fully unknown. Although there are a few sporadic reports saying that some patients have been reinfected after having cured already or recovered from this disease. So all in all, I would say the contamination after recovery is probably remaining for a couple of days, not more. And the fact that the patients that are being immunized and recovering are not subject to additional, I would say, infection is today the basic hypothesis, despite few sporadic reports that some of these patients has been infected again.
That's very interesting, because I think there is a common misconception that if you've had it, there is absolutely no way for you to get it again. So thank you for clarifying on that. Another question is, what is the clinical protocol as far as putting a patient on a ventilator?
Yeah, so that's actually, unfortunately, it varies from country to country, hospital to hospital, but more importantly, it varies on the stage at which you put these patients under mechanical ventilator. I think that's something that we can probably provide in writing as a list of things that needs to be done as a relatively specific and long one with various options depending on the stage at which these patients are.
Okay. A question came in on the, is there any evidence regarding using antibodies from recovered patients?
Yes, actually there are evidence that this might work, but these evidence are actually coming from other diseases. And we do not have, to my knowledge yet, a clear answer to that question, which is the reason why this hypothesis is being now tested in the US, as I mentioned, but also in France. And there is high hope that this would actually be efficient in treating some patients. Nevertheless, we need to consider two aspects. One is the practicality of it. You can imagine that it's not easy to have this done. You need a number of donors and you need a quantity of blood drawing. And secondly, it is also complicated to generalize such an option, even if the study is proven to be effective and correct. So that is why this is probably an interesting option, but definitively not the option for treating COVID-19 patients.
Okay, thank you. Another question is, in clinical trials, what is being used as the standard of care, or does this vary patient by patient?
And sorry, can you repeat the question?
Sorry, the question was, what is being used as the standard of care when being compared in clinical trials, or does it vary patient to patient or trial to trial?
Actually, if we are talking about the standards of care within the clinical trials that I mentioned, it does vary from country to country, but these in general are antiviral. Some uses chloroquines also, and some uses actually immunomodulators. So these are the three categories that are usually done. And of course, as a last resort is the mechanical ventilation and the intubation.
Okay, thank you. Another question is, have you heard of Actemra or tocilizumab being used as an experimental treatment?
I did, but to be very honest, I could not double check the information.
In your opinion, what do you believe is the most promising drug that's currently under development based on the preliminary data?
To be very honest, I think we are at the stage where all antiviral that I've mentioned are more or less having equal chances. We just need to wait a few weeks and then the outcome will come hopefully soon. But I would say, at least from my point of view, the available data does not allow to privilege one option to the others. The only exception I will make is for the hydroxychloroquine, because there has been a really massive talk about that and use of it. And to my personal opinion, so far, it's the hope that are around this are not backed by, I would say, rigorous scientific information. So once again, we just need to wait and a few weeks, and the results hopefully will give the answer.
The next question is kind of like two parts. The drug approval process has been thrown into turmoil. How do you view the FDA's response to the medical and political pressure over COVID-19 therapies? And how has attention to other major illnesses like cancer been affected?
Well, the first part of the question, I think, to be very honest, that FDA is still following a very good way of still, while trying to accelerate such a process of development, of drug development, still using rigorous method to decide how to go about it and how to evaluate these various potential candidates. To your second question, whether that overwhelming request would impact the progress on other disease area, to be very honest, I do not have a clear indicator that it is the case. One might speculate that obviously resources being relatively fixed, that might indeed slow down a little bit the impact for the other development. But I don't think this will be very significant over time.
From a regulatory aspect, in your opinion, what's needed for the current medications that are being tested to get FDA approval in terms of clinical trial results?
Well, as you could see, actually, it depends on the category of the drugs that you are using. If you are using repurposing of the studies, i.e. of the compound or the candidates, i.e. the drugs that have been already approved in another indication, it obviously requires to show that versus either active control of standard care or versus placebo, as you could see from the trial in the US, that you show actually these good standards in terms of safety and in terms of superiority. So I would think that will be the standards, and I don't think that the FDA will deviate too much from that.
Do you believe that one type of vaccine is enough, seeing in regards to mutation rate or antigenic shift, or should the industry really be focusing on looking for more than one type of vaccine?
That's a very interesting question. Actually, there are the various vaccines that I showed you. As you can see, probably the target are not the same. So there are various options and target for these vaccines. Now, that does not mean that this will necessarily answer the second implicit part of your question, which is if we develop a vaccine to the current virus, would that remain actually efficient longer term and even next year? As I said, so far, the preliminary studies does not show a high propensity of the virus to mutate quickly. And nevertheless, of course, if the additional data, which is part of the gray area today, shows that there is a significant mutation over time, that will call on indeed changing that vaccine and readapting the vaccine similar to what we do with the flu. So that's, I think, will be something that we need to follow
on. In treatment, is there any role for anti-sepsis therapies?
Actually, that's a very interesting question. I think with the current situation, I would personally see that there might be some role because, at least symptomatically speaking, these patients go through similar phases. So I would say that this could be the case. I know that some of the approaches are actually considering specifically that roadway.
How can we know if someone is in fact reinfected versus the high level of false negative results? Could it be that they are still actively infected but recovering and potentially still shedding the virus?
Yeah, that's actually an important element. I think there are two aspects to this. First of all, which type of test are you using? If you're using RT-PCR, that will only give you the information whether you have an ongoing actually infection or you just recently had it. The answer to your second part of the question will come from serological actually testing. And usually for that, there are three categories. Most of these recently approved ones are able to target IgG, IgM. So if a patient is having both IgG and IgM, that in general tend to hint to the fact that this patient have had a recent infection and have recovered and started to develop actually not only intermediate antibodies to the virus, but also the most permanent one, which are the IgG. If your patient is having only IgG, it means that in most cases, it means that that patient has actually met the virus, probably had symptomatic or asymptomatic disease and recovered. And that has been some time ago because the mature response of antibodies is there. And obviously, if you don't have any of these, it's clearly that the patient has not met the virus. To the last part of your question, whether they will be contaminating or not, the contamination is actually, as I said, related to how recently the patient recovered. It's not fully clear today, but it is believed that in the early stage after recovery, 1 or 2 days, the patient might still spread the disease, the virus, which it means that if you have both IgM and IgG, you're probably more likely to spread the virus than if you have already IgG because you have recovered since enough time.
One last question. Where do you see the global lockdown restriction guidelines going, dependent on science and potential treatment options to control the infection, or by economics and knowing that further lockdown will hit the economy and global health? Which do you think will influence more? Where do you see it going?
Well, actually, these are, I would say, two kind of acting forces, if I might say like that. Clearly, the economical aspect is not to be dismissed. And over time, this lockdown for a number of weeks, if not a number of months, is definitively making the economical equation more and more complicated. Nevertheless, I think what can help both cases, i.e., the ability to have the deconfinement I would say step-by-step way, would be very much dependent on three aspects. One is the availability of serological tests at a larger scale, so you can understand what is the level of immunity in the general population. If that one is high, up to 60%, then herd immunity will kick in and you're more or less safe actually to deconfine. Unfortunately, the data today are hinting to somewhere around 10 to 15% of the overall population having met and being immune to the virus. So this might not be the better option. Nevertheless, this testing will allow then what we call the search and destroy, i.e. identifying the patient at risk and isolating these patients during the period where they are actually still contaminating. The other general measures are the mask and the availability of mask wearing to the general population. While this is not culturally easy to apply in all countries, and a difference to what is the situation in the US, in the Asian countries, it's still an option that should be, or a measure that should be considered if you stop this confinement stage. And the last one, of course, where the hope would be the most is if one of these repurposing drug that we've mentioned proven to be efficacious, not necessarily to actually prevent the disease, but at least to prevent the progression of the disease and prevent patients from going to this pulmonary complication and therefore to the complicated ICU management needed and potentially deaths. If we can show some efficacy in these currently tested drug, in my view, this would be most likely the highest hope to be able to deconfine in a probably the most optimistic way.
Great. Thank you, Dr. Assia. This has been very educational and very enlightening. We appreciate your time and your input very much. I want to thank everyone who attended today. I hope that this has been a great use of your time. I did want to let everyone know that we will be continuing our webinar series on April 30th. Our topic on April 30th will be coping with COVID-19, maintaining work-life balance. And we'll be hearing from Dan Hennessy, Vice President of Medical Affairs at EMD Serono, and kind of understanding how he and his teams are working to maintain productivity, maintain positivity in pretty radically different working environments. So thank you again, everyone. Please stay safe, please be well, and we will see you next time. Thank you.
Thank you very much. Take care.